Pain plays a crucial role in survival, alerting us to injury, illness, or danger. However, uncontrollable and untraceable pain remains a major problem in modern medicine. Sickle cell disease is the most commonly inherited blood disorder, afflicting individuals primarily from Latin American and African backgrounds. And individuals living with sickle cell disease often experience chronic pain that may arise as early as infancy, and this is accompanied by bouts of extreme acute pain that can also occur throughout life. The current strategies to address pain in sickle cell disease rely primarily on opioids. However, this approach remains suboptimal due to risks of tolerance, abuse, and opioids may even worsen pain. There is a clear unmet need to develop strategies to address pain in sickle cell disease that are devoid of the unwanted, addictive, or potentially dangerous characteristics of opioids. By investigating the underlying molecular and chemical biology of sickle cell disease, we have identified the endogenous cannabinoid system as a potential target to address the complex sickle cell disease pain. Now, what is the endogenous cannabinoid system? As its name suggests, endogenous or endomolecules are compounds produced within mammals, within the body, that can act via various targets. And endocannabinoids are compounds produced within the body that act via similar pathways as the primary constituents of the cannabis sativa plant. Notably, these endocannabinoids are readily absorbed in the body, but they do not produce many of the intoxicating or otherwise undesirable effects associated with cannabis and cannabinoids. But my current work aims to understand how the endocannabinoid system may be involved or play a role in the genesis, persistence, and treatment of pain in sickle cell disease. To this goal, we have developed approaches to utilize the endocannabinoid system through enhancement of pharmacological, nutrition, and behavioral interventions, and to address whether or not pain is being treated in a mouse model of sickle cell disease. To elaborate further on sickle cell disease, it remains the most commonly inherited blood disorder, and it arises from a single mutation in the hemoglobin protein. This mutated sickle hemoglobin forms rigid fibers inside the red blood cells, thus impairing its ability to carry vital oxygen. As well, these rigid hemoglobin fibers change the shape of the red blood cells from their healthy donut-like shape into the crescent sickle shape for which the disease is named. And the reformation and sickling of these red blood cells drives their stickiness. The cells will actually physically clump to occlude or block blood vessels. This blockage completely stops flow of blood, preventing delivery of oxygen and other vital nutrients to different organs throughout the body. The randomness and severity of these occlusive events causes acute bouts of severe pain, often requiring hospitalization. And this can dramatically worsen throughout life and may even contribute to reduced overall survival in individuals living with sickle cell disease. Now, in addition to the physical blockage of the blood vessels, the damage and accompanying loss of red blood cells also contributes to inflammation, which may underlie some of the chronic pain that we observe in individuals with sickle cell disease. However, the mechanisms of chronic pain are still not fully understood. Unfortunately, pain and hence opioid use continue even after curative therapies such as gene therapy with bone marrow transplant, or even the standard of care treatment, hydroxyurea and anti-sickling therapy, neither of which are able to address the pain in sickle cell disease. And unfortunately, over-the-counter approaches to address pain are also carry the risk of gastrointestinal as well as renal dysfunction. So these approaches still indicate an unmet need to address the complex yet delicate pain in sickle cell disease. So to evaluate how the endocannabinoid system may be involved in this, we have utilized the Berkeley mouse model of sickle cell disease This mouse model is unique in that it recreates the physical features of sickle cell disease, allowing us to monitor disease progression over time. And we're able to both enhance as well as deliver endocannabinoids directly to these mice to evaluate its effect on pain. We're able to utilize this model to then combine with cutting edge techniques in high-end microscopy, artificial intelligence, and mass spectrometry to complement our existing expertise in behavioral assessments of pain. These approaches grant us a unique perspective. We're able to now see changes undetectable to the naked eye occurring both within the endocannabinoid system as well as within the larger context of sickle cell disease. To ensure the robustness of this work, as well as our rigor, I have employed a team of multidisciplinary experts from across pain biology, and I am led through this research endeavor with their guidance to help create the most robust research program possible. This also feeds into my own personal research philosophy of inclusivity and diversity, not just of perspectives, but also of ideas and treatments. Having individuals from a wide walk of life incorporated into the lab is a vital component of my research philosophy. So with that, I hope that this work lays the foundation for what will hopefully be the beginning of my own research program to further investigate mechanisms of pain, the role of endocannabinoids in sickle cell disease, while promoting equitable and exceptional research